Thank you, Professor Amengual. Uh, that was a really engaging and informative talk. We have received a few questions. I'll try to take as many as possible, pass it on to you. Are you able to hear me fine?
I don't hear you very well.
Oh, is it better now? Can you hear me better? Is it the same?
I can manage. Go ahead.
Thank you, Professor. So the first question relates to, is there a specific recommendation for carotene or vitamin A intake? And the second part is, could you shed light on the comparative toxicity of carotene and preformed vitamin A when consumed in excess?
Yeah, so that is a very good question because there is a problem with consuming preformed vitamin A. So too much vitamin A coming from, let's say, livers has been shown in the past to cause toxicity. There's reports done from many years ago that people went to when they went to the North Pole, I believe, they were eating livers from some animals with high contents of vitamin A and they died. So there's many reports showing that too much of vitamin A it can be bad for you. It's not exactly the same with beta carotene when it comes to from plant sources, because there are systems in your intestine that regulates the uptake of beta carotene. In other words, if you eat too many carrots, your intestine will regulate the amount of beta carotene that you absorb. You will not absorb everything. There is no such a system for vitamin A. If you eat vitamin A as a retinol or retinol esters from a supplement or a liver, let's say, you will absorb the majority of it. So that is another proof of that eating plant-based meal is a much more a healthy option when it comes to vitamin A for sure.
I think you already answered the second part as well. So there was another question which went like, is there a difference between the toxicity of plant source versus animal source versus commercially available supplementation of vitamin A or carotenoids? So I think you already
So
answered that question.
carotenoids, back in the day, like maybe 30 years ago, something like this, there was some clinical studies that showed that beta carotene could also be toxic when it was given as a supplement in very high doses in a very specific population with already a predisposition to lung disease and exposed to asbestos, because beta carotene is antioxidant. So they thought, oh, well, we'll supplement with antioxidants. It's going to help these people against lung disease. And they saw that actually was the opposite. So in general, supplements of beta carotene are still discouraged. It's not very clear why those clinical results appeared, but definitely supplements of vitamin A are also discouraged, especially during pregnancy. Because as I mentioned at the beginning, vitamin A can affect the development of the embryo. So too much vitamin A, it would be bad. Too little vitamin A is also bad. In general, what is advised is to eat a healthy plant-based diet to achieve the majority of the vitamin A that you need.
Thank you, Professor, for that very clear answer. I'll move to the next question. So since people absorb carotenoids differently depending on diet and genetics, how might personalized nutrition help prevent cardiometabolic diseases?
How what? Can you repeat the second part of the question?
How might personalized nutrition help prevent cardiometabolic diseases?
So what I was mentioning related to the human study, we saw that some people are able to cleave beta carotene better than others. And it can be estimated by doing genetic analysis. Actually, there's some personalized nutrition tests that incorporate the SNP that I was mentioning in my study. It's a very common nucleotide to measure. However, it's very interesting because people that are able to convert more beta carotene in our hands is being shown to be a positive effect. And however, the majority of the studies show that the higher levels you have in the blood, the better from an antioxidant perspective. So there's two parts here. If you have an enzyme that is not very active, you will have higher levels in your blood, which could have antioxidant effects. if you have an enzyme that is very active, you will have lower levels in your blood that will have vitamin A, more vitamin A production. If I would have to pick, I would go for the second option, for the results that we see in our mice and for the results that we saw in humans. The production, endogenous production of vitamin A, because by this enzyme, it has potential and beneficial health effects. That's what we have seen.
Thank you for that answer, Professor Amengual. There is another related question, since carotenoids can act both as antioxidants and as signaling molecules influencing gene expression.
Yes.
Do you think their impact on cardiometabolic health is more strongly driven by direct antioxidant effects or by modulation of pathways such as lipid metabolism, inflammation, or insulin
Yeah, that's
sensitivity?
a $1 million question that we've been trying to argue for years. In my hands, the effects of beta carotene alone they don't have any outcome, any measurable outcome in obesity, liver disease, and atherosclerosis, at least in mice. We constantly see that is the conversion of vitamin A what drives the effects, the positive effects of feeding beta carotene, not the antioxidant. And the way that we can demonstrate that is because we have these mice that, as I showed before, they're extremely yellow. They accumulate tons of beta carotene, but we cannot see differences at the level of gene expression or tissue sizes. The mice, at least in mice, the effects of beta carotene depend on the production of vitamin A. Oh, now I don't hear you. You muted.
I'm sorry about that.
No worries.
So the question was, is the sex-dependent effect of vitamin A synthesis in adipose tissue dependent on sex hormones?
So the effects of sex, we are kind of still exploring why this happened. We know we saw a similar effect in the past, also in mice, but we didn't pay much attention to it because it was a feeding study. This effect is specifically in the adipocyte. This is an effect that happens in the adipocyte because our virus targets only the adipocyte cell. What we think is happening, and we discussed this in the paper from 2022, is that there are the enzymes that go after BCO1. So if you remember from the pathway, BCO1 is the first enzyme from beta-carotene to retinal. Retinal is an intermediate molecule that can either become retinol for storage or retinoic acid. We see that there are differences in sex related to the enzymes that decide whether retinal becomes retinoic acid or retinol. So what we think is that BCO1 partners in a way with in females to drive the flux of retinal towards retinoic acid, and that in males that doesn't happen. When it doesn't happen, retinal is either degraded or formed into retinol. So what we think is that in females, the pathway is directed towards retinoic acid. Yeah.
Thank you, Professor Amengual. The next question is about dietary nutrients. So any specific dietary nutrients that can have either synergistic or antagonistic effects on vitamin A?
We have not really explored much of synergies, but it would be a very good point, because the mechanisms by which retinoic acid work is not only, it works at the level of gene expression and it binds a nuclear receptor. I didn't talk much about this, but there's this other form of retinoic acid called 9-cis retinoic acid. The 9-cis retinoic acid is a very promiscuous ligand for many nuclear receptors, including RXR. RXR, for example, is the partner for vitamin D receptor. So the vitamin D, in cell culture at least, has a synergistic effect when you add vitamin D plus 9-cis retinoic acid. So the effects are synergistically activated. Whether that happens in people or in mice is something that for now is out of our understanding. And there's so many different nutrients, like, for example, fatty acids or oxysterols, or any molecule that is able to regulate gene expression by modulating nuclear receptors, that is makes it all very complicated. We have not really explored that. But yeah, very good question. Thank you.
Thank you. The next question is, you have used both mutant and wild type mice in your experiments. Why not use only wild type mice as model organism for all the studies?
Yes, it's a good point too. Wild-type mice are the ones that produce vitamin A. But when you send these publications with only wild-type mice, reviewers, and that kind of comes back to rigor, we'd always argue, well, is this because of vitamin A or is it because of beta-carotene? The only way to distinguish that is when you use the knockout mice, and then there's no vitamin A in the picture. The use of the two knockouts of the two models allows you to distinguish between vitamin A and beta-carotene. And that is something that, from a mechanistic point of view, is more valuable. Yeah.
Thank you. The next question is, how did you isolate BCO1 and how was it administered to the mice?
And how what?
How was it administered or given to the mice?
The virus. Yeah. So basically, we partnered with a company and also a collaborator that created this vector like a plasmid, basically, and then we cloned the BCO1 cDNA into the vector. And this vector was either targeting adipocytes or targeting hepatocytes because of the promoter that they contain in their structure. So when you inject this reagent into a mouse, these adeno-associated viruses, which are super handy and interesting, they will go into the cell and then will start producing the enzyme. And the production of the enzyme doesn't stop. It goes up as time goes. That's how the adenoviruses work. And there are actually now some of them in the clinical practice that they've been used in humans as well. Not for BCO1, obviously. But these vectors, what they do is to rescue the expression of proteins. And this is how it's used in people that have a mutation. They can recover that functionality by a virus like this. So what we did is kind of the same approach. We had our knockout mice, our full knockout mice, and then we gave them back the enzyme.
Thank you. The next question is, does beta-carotene help in modulating the blood lipid profiles?
Help in the what?
Regulating blood lipid profile or plasma lipid profiles.
Yes, in humans, we saw it in cholesterol, but as I said before, by the conversion of BCO, beta-carotene to vitamin A. There's some studies that show that beta-carotene alone, what it can do is to mitigate the oxidation of lipoproteins, because beta-carotene and all the other carotenoids, which are very similar in activities, antioxidant activities, they travel together in lipoproteins and VLDL, LDL, and HDL, and they help to mitigate oxidative stress. So I told you just part of the information, but I'm not saying that I own all the truth about beta-carotene. There's other things that we typically don't explore. For example, I never measured lipoprotein peroxidation in our mice because my outcomes were different. I was looking at the plaque, not at the lipids as much. Yeah.
Thank you. The next question is, could you shed light on the potential of beta-carotene to prevent or treat some specific type of cancers?
Oof, that is a very, very, very important question. So the way that we see this related to, like what I was mentioning about the adenoviruses, in my mind, what I was thinking is like, well, if we have an obese person, for example, that contains in the adipose tissue a lot of beta-carotene, I could use this vector to produce more vitamin A in the adipocyte alone. Can this be done in the context of cancer? First, we would need to know whether cancers accumulate also carotenoids. Once, if that would be proven, then you could design a virus like the ones that I discussed, targeting those cancer cells, and then induce the production of vitamin A. Problem is that there's an old study, like from maybe 20 years ago, that shows that vitamin A can be very beneficial for some cancers, but it can also be detrimental. Based on the expression pattern of the nuclear receptors that cancer cells have, vitamin A can induce the growth or mitigate the growth of those cancers. So it's a very complicated question. And yeah, first you probably would need to know whether there's enough beta carotene in those tissues. Yeah.
Thank you. The next question is, could you talk about the relevance of studying retinoic acid in biomedical research?
Oh, yeah. So, well, I didn't mention all of the background, but retinoic acid in biomedical research as a whole, vitamin A, I mean, is still in developing countries, is still a big issue. There's a lot of vitamin A deficiency, for example, in many countries around the world. And how that affects the development of cardiovascular diseases is not very covered. How that affects the development of ocular diseases is known. So if you, as a child, underwent vitamin A deficiency for some time, you might have ocular issues and also immune system issues. We in the US, we care more about cardiometabolic diseases because it's the kind of diseases that we experience in the country, right? So there's not vitamin A deficiency in the US, but that doesn't mean it shouldn't be studied. So vitamin A together with iron are the two most common deficiencies worldwide, unfortunately. So yes, I think that in biomedical research is still a very relevant topic.
Thank you for that answer. This is the last question that we have. Although this was not discussed in your slides, or I don't think this is part of your research projects
It's
also.
fine. We will do it.
Nowadays, retinol is being marketed to have anti-aging effects, and it finds extensive use in skincare products as well. Although, like I mentioned, it's not focus of your research. Do you feel there is sufficient scientific evidence to back that claim?
Oh yeah, totally. So I mentioned it briefly at the beginning, but there was like those 4 bullet points that vitamin A and retinoic acid do a lot of things. One of them is embryo development. The other one is vision. One of them that I didn't cover is cellular proliferation. And I kind of said that about cancer. So vitamin A can either induce the proliferation or mitigate the proliferation of certain cell types. When it comes to the skin, vitamin A, what it does is to increase the proliferation of the precursors of the skin. So basically what it does is to produce a peeling. So we will induce the proliferation of those cells fast, and then they will kind of, in a way, rejuvenate the skin. A very good example of this is oral retinoids. So if you have a very severe acne when you're growing up, you go to the doctor and they give you these very strong retinoids. I don't remember the name, the commercial name, but basically it will peel your skin out. The people appear with their very red faces. They cannot be exposed to the sun. They cannot get pregnant during that time if they are a woman. And it's because what it does, vitamin A, is to induce the proliferation of keratinocytes in the skin, and then you peel out completely, and then you regulate again. Problem is, topical vitamin A and also oral is you become very light sensitive. So if you go in the sun with creams that contain retinol in your skin, you will get spots and sunburns. So that can be a problem. Yeah. So yeah, evidence is definitely strong.
Thank you. Thank you, Professor Amengual. Just as a parting thought, any piece of advice that you would like to share with, say, early career researchers who might be there in our audience, or any piece of advice at all?
Oh yeah, so piece of advice when it comes to vitamin A, that would be try to obtain it from plant sources. In general, humans are mostly, we are better off basing our diet in plants than in animals. And vitamin A is one of the many examples that tells you that. As a general advice, career advice, I would say be curious. Try to always ask yourself and other people's questions. Even ChatGPT is very popular now. Don't trust everything it says, but you need to ask yourself questions. While you're working in the lab or you're pipetting, try to stay a little bit away from your phone and listening to podcasts, because it kind of, I see it with my students, it puts your brain to sleep. But when you are pipetting at 10 p.m. at night, working hard, and you have no noise in the lab, you start thinking, okay, what if these results show an increase? What if these results show a decrease? How do I interpret this? So put your brain to work. That's the best tool you can have. That would be my advice.
Thank you. Thank you, Professor Amengual. Beautiful piece of advice and a wonderful and very informative talk. Thank you so much.